Hello, everybody. Excuse me. This is Dr. Sajid, and we'll be having our first online lecture in medical microbiology. Before we move on to the original topic, let me take some time to recapitalize the structure of bacterial cell wall. And if you could remember from the very beginning lectures, that we started with the anatomy of the bacterial cell wall, and we try to find some of the major differences between the two classes of bacteria based on the staining procedures. And of course, we incorporated with that of the cell morphology to broadly define the classification of microorganisms. If you look at this diagrammatic presentation of the cell wall, so starting from the bottom on the right side, you see that we have a peptidoglycan layer. And of course, this cell wall is also having the pili, the organism also having the lipooligosaccharide, of course, polysaccharide capsules, then outer membranes. And if we move ahead on the upper right corner, we have the outer membranes, the cytoplasmic membrane, and in between the outer membrane and cytoplasmic membrane, we have space what we call as the periplasmic space. Now, based on this information, we can decide that this is an organism which is having a periplasmic space. And if you look at the peptidoglycan layer is very thin in this case. So the most probably, when we stain it with the help of Gram stains procedure, this organism will look like a gram-negative microorganism because it has a periplasmic space which is fully defined. It has a less component of the peptidoglycan. So we will talk about a gram-negative microorganism. And this microorganism, it also has a polysaccharide capsule. So this is an encapsulated gram-negative microorganism. Now, when we classify the bacteria based on to the morphological characteristics, so broadly speaking, we have two broad lines, the rods and the cocci. This organism is cocci. So as we have already studied the gram-positive cocci and gram-negative cocci, and we have dealt with that of the staphylococcus and the streptococci, which are both gram-positive cocci. And I'm sure that you know that we will discuss today the gram-negative cocci. And please keep in mind that this organism, which we'll study today, has a capsule, because this is very significant with reference to the virulence of the microorganism, with reference to the identification of the microorganism, with reference to the manufacturing of vaccines of this microorganism. So please remember that we will study a gram-positive, gram-negative cocci, which makes capsules. And the gram-negative cocci, as you know, the major groups includes the family of members of the Neisseria. We have the Neisseria, we have the Moraxella, and the Cetobacter. We will talk about today regarding Neisseria. So as I told you in the very beginning, that all cocci, they are gram-positive. All cocci, whether they're staphylococci, whether Staph aureus, Staph epidermidis, Staph saprophyticus, Staphylococcus pyogenes, Staphylococcus agalacti, all cocci, they are gram-positive except Neisseria. Neisseria are gram-negative, along with that of Moraxella and Cetobacter also. Now, these are the gram-negative diplococci, means that the Neisseria, they occur in pairs. They occur in pairs. And they are having the kidney bean shape. So because they occur in pairs, so we call them the diplococci. Now, one of the other characteristics of these microorganisms that they have this cytochrome oxidase positive reaction. They have an enzyme which is known as cytochrome. Now, this test is very much important because this helps in the identification of the microorganism. We have two important species of the Neisseria, the Neisseria meningitidis and Neisseria gonorrhoeae. As these microorganisms, they occur in pairs, so we can call the Diplococcus meningii, you can call the Diplococcus gonorrhoeae. So we have the gonococci and we have the meningococci. So they occur in pairs. Now this here you can see that the organism, the Neisseria meningitidis, which we also call them the meningococci, it causes meningitis. and it causes another disseminated disease, what we call as the meningococcal. The organism occur in pairs. You can see very well there the organism in pair. They're almost kidney bean shaped, and they are the gram-negative cocci. So the important properties of the Neisseria meningitidis, as I told you in the very beginning in the diagrammatic representation, that the organism has a well-defined, prominent polysaccharide capsule. Now, this capsule is of immunogenic, means that it has the ability to elicit the immune response in the human bodies. And based on to the serological classification, we have 13 serological groups based on to the variation in the capsular antigen. This means that the capsule is not of one antigenic type. It has 13 different antigenic variations. And that's why based on to the antigenic variation of the capsular antigen, we have 13 serological groups. Now, these serotypes, which are based on to the serology, we have A, B, C, Y, W1 to 5. And these serotypes, out of these 13 serological groups, they cause meningitis and meningococcal. Now, the serotype A, which is prevalent all across the world, it's a worldwide in distribution, it causes meningitis. And serotype B, it causes disease only in the USA. The rest of the area of the world is not, you see, prone to this serotype. Serotype B, you see, it is not a component of the universally used vaccine against Staphylococcus, against Neisseria meningitidis. The universally used vaccine against Neisseria meningitidis, it does not have the serotype B because it is not prevalent throughout the world. And this serotype is not that much antigenic. The serotype B is not that much antigenic because it is made up of sialic acid, which is the component of the human cell body. So we have 13 serotypes of Neisseria meningitidis. Most common that causes meningitis and meningococcal are the A, B, C, Y, and W1 to 5. The serogroup A is prevalent in the entire world, but the serotype B is most prevalent in the United States. Now, continue with that of the important properties of the Neisseria meningitidis. It's a gram-negative. We know it. We have to keep in mind the only clinically significant gram-negative cocci are the Neisseria. They have the endotoxin, and that makes sense because the organism is gram-negative. And gram-negative microorganism, they have the lipopolysaccharide cell wall. Now, the exception with reference to that of the variations in the outer membrane of the gram-negative of enteric group and the Neisseria group is that the outer membrane of Neisseria, as we know that it is not a polysaccharide in nature. It's not a lipopolysaccharide. It is a lipooligosaccharide. Now, the lipid A component is common both in case of the Neisseria and in case of the other gram-negative rods, like in case of, you see, the enteric rods. But the outer membrane of enteric rods, which are, of course, the gram-negative rods, is of lipopolysaccharide in nature, whereas the outer membrane of Neisseria is lipooligosaccharide, meaning it has a short chain, it has a long chain of saccharides. So this is the major difference we have to keep in mind. That lipopolysaccharide layer, which is of oligosaccharide in nature, in case of Neisseria compared to that of the enteric rods. Now, the growth of this microorganism, it is inhibited by the toxic trace metals and the fatty acids, so which are the normal component of the bacterial culture media. So we cannot grow Neisseria, both the Neisseria gonorrhoeae and the Neisseria meningitidis, onto the blood agar. We have to use a special kind of media, what we call as the chocolate agar. And chocolate agar is made by heating the blood agar at 80 degrees centigrade. Now, the heating of blood agar at 80 degrees centigrade, it get rid of from the toxic trace elements and the fatty acids, and the microorganism of Neisseria family can be grown onto that. Now, because of preheating the blood agar at 80 degrees centigrade, it changes its color from red to chocolate. So the name for this media is the chocolate. So chocolate is the generalized enriched medium for the growth of the Neisseria, both Neisseria gonorrhoeae and Neisseria meningitidis. This organism, the Neisseria meningitidis, It also has a cytochrome oxidase positive enzyme. Now, this, when you put this colonies which are exposed to the phenylene diamine, they turn the purple or black due to the oxidation of the enzyme. Now, when we place the phenylene diamine onto the colonies of the Streptococcus Neisseria meningitidis, they will change their color because this chemical, it will cause the oxidation of this oxidase positive enzyme. So they will turn into purple or black in color. So this is a way to identify the microorganism. When you have a sample, you have grown it onto chocolate agar, and you just want to identify the organism just being placed in the medium. Will put, you see the chemical, the phenyldimine, and the colonies which are of meningitidis in nature, they will turn their color into purple or black. So this is a presumptive identification of the Neisseria meningitidis. Now, as I said, that the capsule of Neisseria meningitidis is of very unique and unique importance because, as with reference to that of the other capsules, it enhances the virulence because it acts as an anti-phagocytic factor. It does not allow the phagocytosis of the organism. Now, three more important, you see the properties of the capsule antigen are they are defined the serological groups. As I told you in the previous slides, that we have 13 different groups of Neisseria meningitidis based onto the capsular antigen variations. So the capsule, it is an antigen in nature used for the serological grouping. It is an antigen nature which is used to detect the antigen present in the CSF, the cerebrospinal fluid of the patient with meningitis, and this is the antigen which is used in the vaccine. So the scientists, the microbiologists and immunologists, they have exploited this property of the Neisseria capsule to make the serological grouping for the identification and to detect the antigen in the meningitis and to make the vaccines. So very important component of Neisseria is the capsule, which is of oligosaccharide in nature. It's not a polysaccharide. Lipid component is common both in gram-negative rods and in gram-negative cocci. But the sugar components, in case of Neisseria, it's a oligosaccharide, which is a short chain of sugars, whereas in case of rods, it is a polysaccharide, which is a long chain of sugars. Now, the reservoirs for this microorganism are the humans. We are all the reservoir for this organism. We are the natural host. And the organism, it colonizes in the nasopharynx. About 5 to 30% of the civilian population, the military populations, and everyone, we harbor this organism in our nasopharynx. This means that it is the normal flora of nasopharynx area, normal flora. And all those organisms which commonly reside in the oropharynx area or in the nasopharynx area, they are spread through the respiratory droplets and respiratory secretions. Now, this is again, you see the transmission of today's coronavirus, that is COVID-19, that we are facing nowadays, is mostly because of the respiratory droplets and secretions. That's why we'll have to keep mask on our face, on our nose and mouth, so that we can get, we should not get infection with this organism. The organism is transmitted when the carrier becomes in close contact with the non-immunized individuals. Broadly speaking, the infants, they are immunized against the microorganism. The elderly individual above the age of 55, they are being immunized by the vaccines. But all those who have not been vaccinated or they don't have the immunity, they really transmit the disease to those individuals who become very close and correct. Same, you see, true for all kinds of respiratory diseases, particularly which are very transmissible organism and the viral respiratory tract infections. We will have to protect ourselves from the droplet infections and from close contact. Now, the organism from the nasopharynx, it spread via the blood and goes to the meninges or joints, what we call as the meningitis, and it adopted a disseminated mode throughout the body, what we call as the meningococcal. So we have two disease presentation because of the action or the effect of the disease of this microorganism, we have the meningitis and we have the meningococcal, which is a disseminated kind of infection by this microorganism. Now, virulence of this microorganism and the pathogenesis, I'm time and again emphasizing on to the polysaccharide capsule, which is very virulent. It resists the phagocytosis by the PMNs, polymononucleotide, and it does not allow the first line of body immune system to overcome. Now, the endotoxins, because the organism is a gram-negative microorganism, it has a oligopolysaccharide cell membrane, it acts as an endotoxin, and because of the action of the endotoxin, there is a fever, shock due to overproduction of the outer membranous proteins. Now, immunoglobulin A, what we call as the IgA protease, it helps to adhere this microorganism. Now, what do you see? You see, the IgA protease, it causes cleavage of the secretory IgA. So when there is no secretory IgA there, it has been, you see, deactivated by its cleavage or by its component, the immunoglobulin cannot act. So immunoglobulin A protease, it helps the adherence of this microorganism. The microorganism would adhere to the mucous membrane of the respiratory tract by causing cleavage of the secretory IgA, which would otherwise inhibit its adherence to the respiratory tract. Now, the pili and the outer proteins, they are important with reference to the colonization of the microorganism and later on convenience. So, organism enters in the body, it colonizes, it adheres, it colonizes, it penetrates, it multiplies, and it started producing the endotoxin and the other virulence factors which are responsible for the production of different signs and symptoms that we see with this infection. Now, certain people are on high risk, as we have seen that the infants, particularly from the age of 6 months to 2 years, they're more prone to this kind of infection. Now, the Streptococcus pneumoniae and Neisseria meningitidis, they are the cause of meningitis in 80% of the children or infants of 3 months age. 3 months age or over individual to 2 years of age. About 80% of meningitis is caused by either Streptococcus pneumoniae or because of Streptococcus meningitidis. So two major organisms that causes meningitis in almost 80% of the infants over 3 months of age belongs to the Streptococcus pneumoniae, and Neisseria meningitidis. Now, wherever there is, you see the huge congestion of the peoples, it's army recruiters, the hostiles students, the big number of class students, they are the carriers, and this carrier state exceeds over the 90%. So we can see, you see many cases of Streptococcus, of Neisseria meningitidis infection in case of army barracks, in case of hostile students, in case where there are a huge number of students in the classroom. It is second most cause of meningitis in young children after influenza. So we have three important organisms that causes meningitis. We have the Streptococcus pneumoniae, Neisseria meningitidis. Both of these, they cause above 3 months of age, and Haemophilus influenza, both in the infants and in the adults. If you look at the meningococcal disease, there is initially see the organism enter the blood, it establishes, you see the bacteremia, and this bacteremia then goes to, you see the brains and endogiants and become disseminated through the body. And there are two important manifestations of this organism. Once enter the body, establish itself, multiply, bacterial state develop, then there is two implications. One is the meningitis, and the other one is the meningococcal, which is the meningococcal sepsis. So, in case of the abrupt onset of fever, the sore throat, malaise, stiff neck, convulsions, vomiting. So, generalized signs and symptoms are initially seen. Now, in the meningococcal, it's a classical clue is the petechial rashes. If you see small hemorrhages on the body, and the organism is based on the clinical sign expected to have some of the infection with Neisseria meningitidis, The petechiasis due to the endotoxins, vascular necrosis, hemorrhages in the tank and pages. So you can see small blood spots on the entire body, particularly in the trunk area and on the legs and on the arms. So the fulminant meningitis is a life-threatening problem. It has a very high mortality, and this is because of what we call as the Waterhouse-Friderichsen syndrome. Now, the rapid onset of meningococemia, which is life-threatening, is known as Waterhouse-Friderichsen syndrome. There is violent onset of fevers, shivering, chills, widespread spread, you see purpura on the body, disseminated intravascular coagulation, thrombocytopenia, adrenal insufficiency. So Waterhouse-Friderichsen syndrome, which is a fulminant type of meningococemia, it is manifested or characterized by the fever, chills, purpura, disseminated intravascular coagulations, thrombocytopenia, and adrenal insufficiency. Now, how do the lesions of the meningococemia appears? You see, if you look at this slide, we have the petechial ecchymosis, which is scattered all around, the subacute hemorrhages. It can occur anywhere in the body, including the mucous membrane, the saliva, the trunk, the limbs, the arms. And this reaction is a toxin-mediated reaction. So a classical picture after meningitis associated with the Neisseria meningitidis is what you call, this is chemotic or petechial hemorrhages or purpura that you see on the legs. This disease is a medical emergency. We cannot wait for a long time till the results of the isolation of the organism from the laboratory comes. We will have to consider it as an emergency and we will have to immediately start with the treatment. So what we can do in the early stages is that we can stain the CSF, we can stain the blood, and we can see it on the microscope whether the organism is gram-positive or gram-negative. If it's a gram-negative and it occurs in pairs having kidney bean-shaped appearance, we can suspect that the organism is a Neisseria meningitidis. And then we can perform the sugar fermentation test to differentiate between the Neisseria meningitidis and Neisseria gonorrhoeae. Neisseria meningitidis is a maltose positive. It ferments maltose. Neisseria gonorrhoeae does not ferment maltose. So based on the growth of the chocolate agar and fermentation test, the only fermentation test to differentiate between the Neisseria gonorrhoeae and meningitidis is the maltose test. Meningitidis is maltose positive and gonorrhoeae is maltose negative. So we culture this microorganism onto what you called as the thyre martyrism. It is a modified form of chocolate agar in which we incorporate three different antimicrobial drugs to inhibit the growth of the other microorganism and to facilitate the growth of the Neisseria organism. Now, lattice agglutination test is the test of choice, and we can use that on CSF to identify the disease, and it detects the capsular polysaccharide antigen in the CSF. Now, the PCR is also used. So we can go for, you see, the staining procedure. We can culture the microorganism, grow the microorganism onto the methyl martyr medium, or if it's not available, we can go for the chocolate agar, make a slide, see it on the microscope, look for the gram-negative reactions and the characteristic appearance of this organism. And we can perform the lattice agglutination test, and we can go for the PCR. Our treatment is, you see, it's very simple. Once you diagnose or identify the microorganism, the penicillin G is the drug of choice. We can also use the third generation sphaerosporins like ceftriaxons. It has been reported that these microorganisms are somewhat resistant to the sulfonamides, so please don't use that. Recent reports have shown that this organism is becoming resistant to ciprofloxacin also. So please avoid not to use the ciprofloxacin and sulfonamide, but keep your options open for the use of penicillin G and third generation cephalosporins. Now, if you look at the prevention of this disease, which is really important, the chemoprophylaxis and immunizations both are used to prevent the meningococcal infections. We can also use some of the drugs like rifampicin or ciprofloxacin as the prophylactic drugs. We have vaccines, and the vaccines against the group C and Y and W135, uh, they contain the polysaccharide capsules. And the vaccine against group B, meningococcal, contains also a factor which is known as the H binding factors for this region. So the vaccine is highly recommended. It's freely usable. It is safe, and we can use these vaccines in both areas. Uh, there is a dire need to use these vaccines as a prophylactic uh, to overcome the incidence of Neisseria meningitidis, which causes meningitis and life-threatening meningococcal in case of uh, adults. That was all about the Neisseria meningitidis, and we can go for the lab techniques, what you call it, the oxidase test that is used for this purpose. You know, you see the colonies and all that, but it's not that important. I have already gone through what we have already seen. So that was all about 2 days lectures. And I hope you enjoyed your journey of learning online. And we will meet again and we will discuss the other important organism, which is the Neisseria gonorrhoeae. Thank you very much.